Hi friends, today's video is about gram staining. This staining technique was originally developed by Hans Christian Gram in 1884. So let's go to the procedure. The first step is fixation. So the smear is made on a slide from bacterial culture or a specimen. It is air dried and then heat fixed. Next, we are going to add the primary stain. The smear is stained with para-rosanilin dyes such as crystal violet or gentile violet or methyl violet for 1 minute. Then the slide is rinsed with water. The crystal violet stains all bacteria violet in color irrespective of whether they are gram positive or gram negative. So here we can see the application of crystal violet. So the all bacteria are stained violet in color. Next step is adding the mordant. The mordant we are adding is grams iodine. It is nothing but dilute solution of iodine. It is poured over the slide for 1 minute. It is then rinsed with water. The mordant is nothing but a substance which helps in fixing the smear onto the slide. The next step is decolorization. We are pouring a few drops of decolorizer to the smear. For example, acetone, ethanol, etc. can be used as decolorizer. The slide is immediately rinsed with water. The decolorizer removes primary stain from gram-negative bacteria while the positive bacteria retains the primary stain. So here the alcohol wash or decolorization we can see. So the gram-positive bacteria are still violet in color. So they are retaining the primary stain, while the gram-negative bacteria, the primary stain gets removed from that. Here, the decolorization is said to be the most crucial step. Why? It's very easy because if we are pouring it for a more time, even the gram-positive bacteria loses its color. So, it is over decolorization. And if poured for less time, the gram-negative bacteria do not lose the color of the primary stain. So, there will be under decolorization. Hence, it is called the most crucial step. Okay, the next step is counter stain. Counter stain means secondary stain. We are adding secondary stains such as safranin or diluted carpal fuchsin for about 30 seconds. It imparts pink or red color to the gram-negative bacteria. Okay. So the slide is rinsed in tap water, dried and then examined under oil immersion objective. So after adding the counter stain, so here we are adding the safranin. After its application we can see the gram positive bacteria still retaining the color of primary stain, that is violet. The gram negative bacteria got a pink color, which is the color of the secondary stain. Okay, so interpretation. The gram positive resist decolorization and retain the color of primary stain, that is violet. While the gram-negative bacteria are decolorized, they take the counter stain and appear pink. This is the interpretation. Finally, going into the principles of gram staining, though the exact mechanism is not understood, the following theories have been put forward. First one is the pH theory. So the cytoplasm of gram-positive bacteria is more acidic. Hence, it can retain the basic dye for a longer time. So, the crystal violet we are using here is a basic dye. So, it can be retained for a longer time. Second one is the cell wall theory. The gram-positive bacteria has thick peptidoglycan layer and they are tightly cross-linked to each other. The peptidoglycan itself is not stained. Instead, it acts as a permeability barrier, preventing the loss of crystal violet. Also, the alcohol is thought to shrink the pores of the thick peptidoglycan. Hence, the crystal violet cannot be easily moved out of the gram-positive bacteria. While the gram-negative cell wall is more permeable, thus allowing outflow of crystal violet easily because it has thin peptidoglycan layer and also there is presence of lipopolysaccharide, which is exclusively present in gram-negative bacteria. So this lipopolysaccharide gets easily disrupted by the acetone or alcohol, thus allowing the primary stain to come out of the cytoplasm. So this is all about gram staining. Hope you understood and thanks for watching. If you find the video useful, kindly like, comment, share. And please don't forget to subscribe. Thank you.